Thank you everyone for being here. Actually, outside was very nice, right, today. So, okay, today I'll talk about the project, the topic I'm talking about is sulfoproteomics. So mainly I'll just talk about what is sulfoproteomics, what is the current, challenges, and I'll introduce the real application in Golgi cell cycle. To talk about sulfoproteomics, I just want to go over first of, just a general biochemical, like a surfactant reaction that is found in cell, and then, and a few examples, and then go briefly review what is the surfactant and what is phosphorylation as a post-transfer modification on proteins. And then introduce the sulfoproteomics workflow that I applied to Golgi cell cycle, and how high resolution mass spectrometry and delta mass analysis is used for protein surfactant analysis. So here, sulfur, I just showed the picture in the title slide. There shows like sulfur mine and the hot springs. Sulfur is actually top 5 most abundant element of Earth, like by mass. So naturally, the organisms evolved by using the resources that are available in the environment, right? So sulfate is because in aerobic environment of Earth, the highly oxidized form of sulfur is a sulfate. So sulfate is readily available, but in biology, this PAPS is the biologically active form of a sulfate donor that is used for all surfactant reactions in cells. So I'll just show the general view of how surfactant is occurring inside of our cells. First, the sulfate. Oh, no, okay, yeah. Okay. Okay, this so the sulfate from sulfate is readily available through the metabolic process by microorganisms in soils, and then once the inorganic sulfate enters inside of a cell, like human cells, they convert it to biologically active form like a PAPS by enzymatic enzymes activity. It's a dual function enzyme called the PAPS synthase that is located in cell nucleus or cytosolics. So then the sulfate from PAPS is transferred to other endogenous biological molecules, like such as a lipid, carbohydrate, or proteins. By general, like a group of enzymes called the sulfotransferases. So I'll just in this slide I'll show one example of one of the abundant surfactant reaction that is happening in the biomolecule. It's a lipid sulfation. So lipid is everywhere, right? So it's a composing component of our body, or mostly highly populated, like our skins or brains or arteries, all that. After all, the lipids are also like a certain portion of the lipids are sulfated by sulfotransferases in cytosols. So one of the example is it's biologically significant is cholesterol sulfate. Cholesterol sulfate is most abundantly found in like a fluid system, like a plasma, urine, or bile, like that. And then it has been known that the concentration of sulfate cholesterol in blood is like strongly correlated with pathological conditions such as inflammation or neurodegenerative disease. So it could be, the sulfate cholesterol can be a biomarker or diagnostic, factor for those kind of pathological conditions. And also, it's been well known that cholesterol sulfate is involved in blood clotting and fibrinolysis. It's a reacting with key player proteins that involved in like a blood clotting and fibrinolysis. Okay, so another very interesting example of a sulfation on macromolecule is GAG, the glycosaminoglycans. Maybe GAG or other like polyglycans are actually one of the most heavily sulfated molecules in our cell. So and then and also it's GAGs are mostly present found in extracellular matrices, like it has a long chain of polysaccharide attached to the core protein. It's like building a fuzzy protection layer outside of a cell. So it communicates with the extracellular signaling or preventing virus infections such as that. So, and then very recently, there's a very interesting study has been presented that chondroitin sulfate. It's a type of GAG polyglycan, and then it has been known that the length, how large of this polyglycan, and what kind of GAG is the dimer is taking, and it's modulate the interaction with other molecules. But this group in Germany, they reported that only one specific sulfation coding.
I can help you. It's all right.
Okay, so it's not. Okay. Yeah, I can just
See, now here it is, right there. It's in there. Now, if you hold that top one down.
Top one, okay.
Oh,
Oh, okay, now, so, yeah, only this a very specific modification of sulfation on free hydroxyl group, only this particular coding can activate the growth factor in brain. So not other. So if you think about it, because if this tetra polyglycan is just one, but by changing the coding, it can upgrade to 256 different ways of coding. So it's this is a syntax word, from one word can evolve into 256 different ways, like a full communication can be evolved. So it's a very interesting, but it's still very early because the technological, the challenges, because it's still very, there's no technology, is available to provide its molecular level structural details. But the high resolution mass spectrometry is rapidly evolving, and then I think in the near future, I think a lot of studies will come here to explain how this sulfation on GAG will change their interactions. I think it will be very exciting to see all that. Okay, next one I'll just talk about is protein sulfation, the main topic that I'm going to present today. So protein sulfation is also so the paths that the paths from inorganic sulfate and then transfer to paths the biologically active form is transported to Golgi in the cell, and then there's a Golgi enzyme called tyrosyl sulfotransferase. It's a specific enzyme that modifying protein tyrosine site here to sulfated tyrosine here. So there is only two type of TPST enzymes found so far in human cell, like a TPST1 and 2. They have like, sequence is very similar, but their specificity, the subsets are different. So it has been known by genetic analysis here. So, and then so far what's known is the sulfated protein, mostly sulfated, and then because of the sulfation, it's secreted to outside of the cell, or they located to the plasma membrane. So even the implication of the importance of the sulfations, that involving the lots of human health and the disease, and also like extracellular signaling, but sulfoprotein has not been much like studied because of its technical like challenges that we have, limitations. So this is the infogram that I found on online, that some review paper, and I think it's really well depicts how what's the current status of the sulfoproteomics. So the big circle on the top is the number of sulfoproteins across all across all genes or species. Only like less than 500 is reported as sulfoprotein, but experimentally only 148 are identified as a sulfoprotein. And then that numbers dropped in human. Only 32 human proteins are reported and they're characterized as a sulfoprotein. And then the 32 is, or 14 of 32 is a cell membrane, and then the others are secreted proteins. And then there are molecular function and biological processes. It's, I already a little bit of explained that they're mostly involved a lot of, functions and like process of blood coagulation and like, let me see here, like fungicide, the apoptosis. It's like it involved in a very critical biological processes. So so why? Because why it's like the sulfoproteins are like heavily or severely underexplored is the reason, because the sulfation is naturally, by nature, it's very labile. Under acidic condition, it just formed protonated and then like spontaneously dissociated into sulfate. And then also it has negative charges, carries negative charges. Okay, here. So that means it's a very unfavorable for mass spectrometric analysis that we're using in typical proteomic workflow, because all proteomic workflow based on LCMS developed on positive ion mode electrospray LCMS. And then in positive ion mode, they don't really charge well. That means their ionization efficiency is poor, detection sensitivity is low, and then also in mass spec analysis, the SO3 is fragmented. The locational information is lost. So, and then also another big reason is it's competing with a very abundant phosphorylation. It's the same like a negative charge, like providing negative charge to protein, like a sulfation. But it's phosphorylation is much abundant in cell. So this is just a table that comparing the similarity and the differences of phosphorylation and sulfation. And phosphorylation has been really well studied, especially the last 2 decades since we and then for now I think over 500 kinase has been known in cell, and then people say roughly 70 or 80% of all proteomes are phosphorylated at least once in their cell lifetime. And then because the phosphorylation is a reversible reaction by two enzymes like a kinase, putting phosphorylation kinase and phosphatase. So it's basically phosphorylation work as like a molecular switch to turn on the activity and turn off very quickly, efficiently. So that's why, the biology evolved utilizing the phosphorylation in a lot of processes, including, the signaling, all that. So it's not surprising that a lot of like drugs, like those are therapeutics are targeting the kinases, that reason. But in the other hand, the sulfation has is sulfated by sulfotransferases, but no sulfatases has been reported in human or high order like virus. And then it only about at around 20. I just showed those, right? About 200 proteins are sulfated in like a secreted and membrane proteins. And then it's been known like they've been involved in mostly like extracellular signaling. And then these the chemically both negative charge is similar, but also mass spec way, their masses are pretty close, like only 0.0095 Dalton differences. So it's a very challenging to distinguish those two isobars. We call this isobars, yes.
Is there a way during the sample processing preparation that you can like chemically modify the sulfation group so that like it's not associated with phosphorylation anymore or ionize better?
Yes, that's a good question. And the people actually has, studied, been studied all those of how to protect those ones for phosphorylation as well. Phosphor also, when you started the project, people say phosphorylation is a labile because under this condition, like acidic condition and the mass spec condition, they're just very easily dissociated. So people come up with, okay, first block chemically modify all free the hydroxyl groups like that, and then leave only the modified one. And then when you cleave, it's still there was, there are other ways that chemically you can modify and track the phosphorylation site or sulfation site. But that's for targeted studies still can be valuable, but in large scale with a very complex sample, it's just not practically, working that well. So, because proteomics means with a large set of complex sample in limited time with a limited instrument, so you want to have as much information out of it. So that, then that kind of chemical modification, if it's not like proved to be like 100% like conversion ratio, then it's not practically work. So that's why this protocol has been adapted for phosphoproteomics. So, and then this has been so it's already established for phosphoproteomic analysis, and then with the mass spectrometry, and so adapted by a lot, globally for most proteomic lab working on phosphorylation. So it's a 3-stage. First, the sample preparation, and the LCMS, and the data analysis. In samples preparation, first, proteins first digested, and the peptide, and then phosphopeptide usually very low in overall, because you want to have enrich the only phosphorylated peptides by using their chemical property, right? So, and then in early 2000, we found that there's the method, the selective enrichment of phosphorylpeptides that by using metal iron IMAG or zirconium and titanium dioxide, because this phosphate group is a polyprotein, has a lot of electrons here, so they act like a Lewis base on the vacant metal, positive ions here. So it has been shown that these techniques can really selectively enriched phosphopeptides among other, non-phosphorylated peptides. So this selected phosphopeptides, enriched phosphopeptide, separated LC electrosprayed, and then analyzed about high resolution mass spectrometry like Orbitrap, FTIR, because these new modern instruments provide really high resolution. So, sometimes you can distinguish, of course, like distinguish the isotopes, even, distinguish like a 1 electron differences in where highly, well-defined conditions under condition. So once the peptide mass is accurately measured in MS, and then the next stage, the peptide is going through fragmentation by applying, energy. So currently, these kind of fragment, like tandem techniques available. So CID, TD, and UPD, these are different by the reaction mechanism. So that means their reaction different, they generating different fragments. So they can be used to complementary information. And then once the data from LCMS, like MS accurate mass one, and the MSMS from like sequence information, can be submitted to database search, to match with the known the sequences of like mouse like protein from Uniprot or NCBI, and then matching, and then the database search algorithms like MaxQuant or Mascot or other search engines providing scores for how well the spectra matches to the sequence, and then the search results can filtered by FDR, and then the final, the information will processed to pull out the biological information by using many other like the bioinformatics analysis. So to come up with a sulfoproteomic analysis, I said already, sulfation is really labile. So we have to, so because of those kind of challenges, like it was like even 2 years ago, the general norm is large scale real proteomics for protein sulfation is not feasible because of their just already, before detected that they lose sulfation information. And so it's a conventional like workflow. They don't really work for labile, like PTMs, sulfation. And then back then we didn't have the selective enrichment method was known. So, but so now, so that's why I've been working on phosphoproteomics for a while. So, and then by thinking of the chemistry of the phosphate and sulfate, both negative ion, both highly polyprotic, anion. So, and then the way they interact with the metal ion, I thought that maybe should work for sulfate. So I tested with simple mixture of like not like a sulfopeptide or phosphopeptide with other, and then tested on the conventional phosphopeptide like enrichment like techniques with IMAG and titanium dioxide. And this is before enrichment. You can see those phosphopeptide here, and also sulfopeptide gastrin here are like very common, like a standard sulfopeptide, CCKS peptide, also sulfopeptide, co-enriched with a phosphopeptide. Well, their efficiencies are pretty much similar, but there's, if you can see, their specificity different by different enrichment technique, IMAG or titanium dioxide. So, and then also, this is enrichment. And then when you go to look for the location information, to tackle the labelability, in the like ECD or ETD is called like electron-driven, free radical driven, like a soft fragmentation. Their studies has been known that these two techniques are like preserve labile modifications like phosphorylation and sulfation and also like glycosylations. And then we saw that a well-controlled ECD and ETD experiment can preserve the very labile circulation as well. But the problem is this type of fragmentation, the tandem techniques are taking longer reaction time than CID or HCD, and also the fragmentation efficiencies are lower. So after all, for the high throughput, large scale like data analysis experiment, they don't return good, IDs result. Also because we're using on a lot of the search engines, available, but the publicly available engines are not really well tailored for this ECD and then ETD based like a fragment. Because all majority of communities are using like CID and HCD for B and Y type of ions. So the current available the algorithms are not so well working with that one. So still, so people use this one, but it's not really like a primary tool as for the large scale. So okay, I'll just move now the study that I have done that I applied the sulfoproteomics. So it's a Golgi, so okay, so Golgi here. Golgi is it's a membranous organelle in cell, and then because of this distinctive shape, like a stacked pyramid, like right. So because of this, it's very easy to find in the cell. So if you see the cell diagram, you can see where the Golgi is, right? Golgi is like a portal for protein sorting and the protein modification. So once proteins are synthesized from RNA, DNA and RNA, they go to Golgi, and here they sort it out, and then they put the modifications here, and then sorted, transported to their final destination. They're secreted or other part of cells or body. So that's what they do. But actually, the Golgi is not just always this shape. During the cell cycle, like cell division, division, the Golgi actually disassembled into smaller fragments like that. And then once the cell division is done, it's split into two daughter cells and then grow into full like a Golgi cell shape. So this very interesting like a biogenesis cycle, my collaborators are studying, but the molecular mechanism of the Golgi cycle has not been known. So that's our study. So we want to study, what proteins are involved in this process and then what kind of PTMs are, involved in this process. So that's how we started the project. Oh, yeah. Okay. And then, so this is the sample. So the Golgi during interphase. And then we collected Golgi from mitotic state, the Golgi fragment. So we compared two sample set of Golgi through this process. So the process is, I got this from like proteome, phosphoproteome. So first, sulfopeptide sequence enriched by different technique here, IMAP, and zircon is sequential because they have a different specificity. So I want to, benefit from that. So that, and then they ran separately on C18, orbit LC, orbit trap, and applied HCD as a tandem technique by using 30%, collisional energy. And then, so the search engines conventionally use like a max quant or sequest. When you do search, you specify, you're given the parameters, what you're looking for, like give the specific PTM informations and then like that. So, but usually those are, we've been known that this kind of, this conventional search engines does not really well work on label modification because sulfopeptide, there's no evidence of the modification is present, then usually those kind of peptides misidentified or as like a phosphorylated peptide or unidentified, even if the quality is good. So that's what we've been observed. So and then at the time, like a lot of, in like bioinformatics, the studies has been like, people presented the open search concept, and then different tools were available, like open MS Fragger, open PFind, because these open searches are very powerful for especially unknown modifications, because these are not necessarily, limited the peptide to look for like certain modifications. So yeah.
Yeah, so why did you use open search instead of variable modification search just to find like phosphorylation information?
Why? Okay, so I tried different, like conventional tools, but usually they don't really come up with a good ideas very well. And then, the open search, when it's introduced, it's a big concept is looking for unknown modification, also label modification. So that's the initiation. Okay, let's try open search to see whether this is picking up the label modifications. And at that time, it was not really evaluated for those kind of label modifications very well, very early stage. So also the purpose is to compare, these different algorithms, how well, picking up and what's the differences. So that was one of the purpose. So I just the data sets submitted to 3 different search engines, and then we validated the sulfopeptide of PSM. So like a lot of manual validation work has been done to validate the IDs, or also some rational comparison of 3 different algorithms. Okay, so delta mass. So I think it was because you're from mostly like bioinformatics, so I think you're familiar with MS Fragger and then that, but so I'm not going to really detail. Actually, I don't really know in detail how they work, but anyway, delta mass is an algorithm embedded in MS Fragger, the open search. So what it does is MS Fragger first wide open, like plus minus 500 Dalton differences, and based on the fragment, it finds the sequence, and then it reports the mass difference, the sequence-based rebuilt the theoretical mass to the actually measured precursor ion mass. So the delta mass usually match to the modifications, like a protein modifications, if it's like phosphorylation. So it has so if you plot the profile, like the mass differences on a number, this is a very narrow region, but if you here, you can clearly see there's oxidation, comment, all that. It's a very a great way to see the data in the context of PTMs and modifications like that. So we found, so in our sample, like interphase Golgi and the mitotic Golgi in the region of sulfation and phosphorylation, we found the sulfation and the phosphorylations are clearly separated. Like it's, there's no overlap. It's clearly separated. So I think this is really striking. This is this is my personal opinion, but delta mass is the best way depicting high resolution, impact here. So because this isobar, and then the mass of center of this profile observation is centered exactly at the theoretical mass observation, and this is exactly centered at phosphorylation. So there's no doubt that these are ambiguous. So clearly, right? And then, so another one, so when I take in from this two separated, profile, I pull out the sequences. When I take it, so I only taking the 2 standard deviation empirical rule, and then when I pull out the sequences of phosphopeptide and the sulfopeptide from this, they don't really overlap. So it's a, because sulfation, kinase, and sulfoperoxide, they have a different substrate. They serve different like biological functions. So it is expected, but and then seeing the real result and experimental data, it's very exciting. So it's clear. It also prove that that this kind of delta mass analysis can allow identify and distinguish sulfation from phosphorylation. So I think it's very I was very happy with that result. And then so after all three algorithms comparing, like MaxQuant, pFind, and the MS-Frager delta mass. So I compared the peptides and then the Venn diagram, and then large portion of it actually overlap, like a 35 of that. And then, but if you see here, MaxQuant, so MaxQuant and the OpenPFind finds a lot more like a sulfopeptide than MS-Frager. But after manual validation, like then a lot of the OpenPFind and then MaxQuant IDs are filtered out because they're not sulfopeptide. So after that, I feel like really strongly that maybe the delta mass by MS-Frager can be a very powerful tool for this kind of highly labile modification study. So, but there's still limitations for those tools, because it only reports by delta mass. So it's hard to get quantitative information other than spectral counting. So that's one drawback. And another is still, we don't know about the location of sulfation because there are just already lost during the fragmentation. So this is summary, what I found from the analysis. So I reported here, we found the overall 26 sulfated protein. Think about the current, like before, only 35 was analytically identified human sulfoprotein. I added 23 new proteins in that. And the three of them actually has previously been the red one, fibrinogen here, serpin, and the TPSG one has been already reported as sulfated, but we found that one. And then later, after our publication, these 4 blue ones are also found in other studies. So also, and then also another thing is we found the sulfation site here, like because luckily those peptide has only one sulfation in single peptide, so we could localize that unambiguously. But if it has a multiple modification site, we can't really pinpoint the location, so it's a little ambiguous in this domain. So that's why. But overall, we have 30 site localized sulfopeptides. 30, yes. So okay, so here. So just to show some like actual data, how the high resolution mass spectrometry can distinguish, the close isobaric phosphopeptide and sulfopeptides with some examples. So one of the secreted protein we found here is with the sequences like that. So we found in one peptide had a sulfated on this tyrosine site, and then in one case it is also phosphorylated in this serine site. When I pulled out the spectra of this particular modification, so this is Y9 sulfated peptide here. All these fragments I have marked with a star mark is it lost the loss of sulfation. So none of these fragments has the the modification. So we completely lost the site determining, PTM information here. But in case of it's a phosphorylated, the same peptide with a phosphorylated on serine, serine here, the peptides retain, okay, so, okay. Oh, not, sorry, no, not retained, sorry, I was confused. So the serine is also lost, but the the phosphorylation neutral loss is not 80 Dalton loss, it's a 98 Dalton loss, because it's losing a neutral H3PO4, the mass is 98. So the loss is 98 from the fragment. So I marked it with a pound sign here. So if you see the peak corresponding, its peak is about different by 18 Dalton. So that's how this one, clearly this tells that this is a phosphorylated peptide. Or even we can have the localization information, but at least this we can tell. This is phosphorylated peptide. This is sulfated peptide. And also these two isoforms of sulfur and the phosphorylated peptide separated by LC gradient here, like on LC. So the red one here is sulfopeptide, and then the mass, you can see the mass is also slightly different. So the blue one has slightly higher mass because of the precursor ion loss coverage. So this is, so in the data set we have, we found something we didn't really expect to see. They said phosphorylation and sulfation, they are chemically similar, but biologically they serve really different, processes. So they interact with different substrates. But here we found some peptides has both sulfated and phosphorylated at the same site. So that wasn't really expected. So we thought it's really interesting biologically, also analytically. So if you see here, this A1I3 peptide here, and then sulfopeptide, of course, here, all these peptides, there's no sign of modification, all complete neutral loss. But when it's phosphorylated on tyrosine, the same site here, tyrosine surface, because tyrosyl phosphorylations are chemically, more stable. So they have these fragments retain the phosphorylation. So by this spectrum, this peptide accurately localize the phosphorylation site on this tyrosine. Another example, interesting thing is EBAC9 peptide here. This peptide is one phosphorylation and one sulfation in the single peptide here. So the phosphorylation site can be localized by this, phosphate retaining fragments, but sulfation is the site is lost. But another case, the same peptide with this one is a doubly phosphorylated, and this is 3S and then 15 tyrosine site is doubly phosphorylated. Because it's phosphorylated, there are all these fragments that carrying the modifications, so allowing us the localization of both phosphorylated sites here. So it's a protein crosstalk. So so this wasn't really we're looking for in our study, but this probably the first evidence that phosphorylation and sulfations are competing and for the same site. It's like reciprocal crosstalk. So, so it's still like a speculation because we don't have a biologically, like biological evidence or like that. But because the sulfation I said is mostly because the TPST, the enzyme is Golgi resident, Golgi site. But most of the phosphorylation kinases are in cytosol. So maybe, you know, the proteins are, once it's a phosphorylated in cytosol, they will not be sulfated. Also, it's a wide, you know, like speculation. Maybe it's sulfated because this side, this phosphorylation will not, because this sulfation is similar to chemically, it will act like antibody for phosphatase working on this side, so making this phosphorylation biologically active. So those kind of, you know, like speculations we could make it. But so far, this kind of rare crosstalk between phosphorylation and sulfation, we haven't, you know, found other than this study. So it's still, you know, future study is warranted. It'll be interesting. Another, this is not like a phosphorylation sulfation, but in other study, like the crystallography study and the immunization studies, it has been reported that TPST enzyme in Golgi is present as a dimer form. So sometimes a homodimer like a TPST1 and 1, or sometimes it's a heterodimer as different TPST1 and 2, or in some cases also making heterodimer with the glycosylation enzymes here. So that means, you know, the sulfation and the glycosylation has, you know, high chance that regulate each, you know, activities or, you know, modifications, like potential crosstalk is, you know, suggested here. And then, you know, it is not surprising because this is the proteins that we found in our data set. So it's a protein interaction maps, but a lot of these proteins, the sulfoprotein we identified is actually involved in like a sulfotransferase working in like a glycans. So, and then a lot of them are actually the glycopeptides. So potentially they're crosstalks between sulfations and glycosylation. Okay, next one. So this is the last slide from the data. So even the sulfation was lost, but because of the only one tyrosyl site is in 30 sulfopeptides, so we could localize those ones. So we aligned those sulfation sites at the center, and then just to compare which amino acids around that sulfation site. So you can see, you know, the -1 position of tyrosin's sulfation site is like largely populated by acidic, like a D and an E. So it consists with our knowledge that, you know, before the sulfation is mostly found around acidic moiety. So it's a well agreed that one. And also, you know, other than the acidic, there's a not really specific motif. Is like a phosphorylation has a very, you know, each kinase have a very specific like a sequence motifs, but this sulfation does not give that specific motif other than this generally acidic, you know, moiety. Okay, so so first, you know, so I talked about, so, you know, I just want you to at the end, you know, just to take away the message that what is sulfoproteomics, and then And then what other, surfactant reactions in biology, not just in the context of sulfoprotein, but it's a surfactant widely interacting with the different surfactants in biomolecule. So I think this, because of this kind of surfactant pathway has been like really extremely like understudied because of those technical challenges. But now like mass spectrometry really like evolving really like fast, and all those available informatic tools for that. So I think there will be a lot of work that can be done on this surfactant in general, also as well as the protein surfactant. So and then let me see. Okay. And then, okay, I didn't really emphasize that during the slide, but so in the in the 4G mitotic and interphase, I didn't really point it out. The phosphorylations are really high regulated during mitosis, but the surfactant is remained unchanged. Well, so that means phosphorylations are like plays in regulating factor of the 4G cell cycle, but surfactant is not involved in cell cycle. But other than that, the surfactant side is really well regulated. So so that means TPSD1 and 2 it's not involved in cell cycle, but it's a highly regulated processes. Okay, so that's all for today, and then thank you for your time here today. See if you have questions, yes, please.
So have you tried to like incorporate like TNT to see, because lots of the cognitive study will use the TNT to probably more like in some way or like reduce the like trying to quantify different like different segments, right? They have you tried to, because it looks like you didn't change much of the protocol between the possible enrichment and the
Therefore, yes.
yeah, sulfopeptides, right? So I just wonder, have you tried like TNT? And at the same time, probably there are additional question like, what's the HCD energy you have tried to still see the signal?
TMT, TMT is like a widely used for quantitation, or also with the phosphorylation as well. But because TMT is the mechanism, the quantitative, it's because it's it's cleaving the reporting group, right? So that means in mass spectrometric aspect, TMT labeled peptide with a labile modification is actually it's a competition for the energy, the collision energy. So if it's phosphorylated, the energy is not going to make the reporting group of the TMT for quantification. It just wasted to more labile phosphorylation. So so you have to be a little cautious when you're using TMT for phosphoproteomics. Nonetheless, the surfactant is much labile than phosphorylation. So it will, I think the TMT will give you quantitative information for protein level, but this is not, warrant for the surfactant quantitation at all, right? Because surfactant already lost.
But
Yes.
like you said, like glycosylation, those things, I think TNT also being applied to see like co-enrichment of like glycosylation peptide. So I think that's also possible, right? I'm just kind of thinking that, because for TNT, they are saying we don't change the positive and negative mode, but if you just search the possible enriched TNT data, you still find the glycosylation. So I just wonder if it's possible to find those things in other data sets. Or probably you can mention like what kind of protocol changes you have done. Do you do positive and negative mode at the same time right now, or you just still use a positive mode?
Mostly, use positive mode. When negative mode, surfactant could be stable in negative mode, but it's not in general community. Negative ion mode analysis not well supported by tools of that. So, there are certainly people's working on that niche to that, but it's just not popular yet. And TMT is I think it's a little different. You know, with TMT can be applied for quantification, but it's not really overlapped with localization information of label modifications. So, yeah, so, okay.